Chapter 6, Section 8, Transfer RNA. Transfer RNA, more commonly referred to as tRNAs, okay? These are the RNAs that, well, basically are chaperones for the amino acids. They bring the amino acids to the translation party. The tRNAs, each of them are going to have a anticodon, a 3 RNA nucleotide sequence at the base of it that will interact with the corresponding codon on the messenger RNA. So that way you get the codon anticodon. They call this this complementary base pairing. This is the way to confirm that the cognate or correct amino acid is the being brought in and will be the next one added to the growing polypeptide chain. Prokaryotes have about 60 different tRNAs. It turns out that, there's going to be some redundancies in these tRNAs. Certain ones are going to only be used under specific times. We humans have about 100, 110 different ones. As always with the RNAs, they're single-stranded, but the thing is they are not just a straight line. They are going to complementary base pair, bend around on itself, forming these hairpin loops and bulges and things like that. So it gets extensive secondary, three-dimensional structure. See if I got a picture of it. This base pairing, they call it a clover leaf shape, but you have the clover leaf spread out and then imagine it hugging itself and bending. That's the overall shape. On the left here, that's the clover leaf that you'll see drawn in many textbooks with the arms splayed out there. Down here at the bottom, here's the anticodon, which is going to be the complement of a codon that's going to be on the messenger RNA. 3 nucleotides, 3 nucleotides. Up here at the 3' end, this is where the amino acid will be added. So this is a charged tRNA. Charged meaning it has the correct amino acid attached to it. This is honestly more along the way what it looks like in a ribbon model, not a space filling model, just the ribbon model. You take the clover leaf and like I said, you bend, wrap the arms around itself and then bend over. There you go. That's what this is. It's actually this, but then just, like I said, wrapped it on itself. Amino acids are activated by ATP to form an amino acyl AMP. The AMP is then going to become a charged tRNA because there's going to be an amino acyl tRNA synthase. It's going to come through and it's going to add on the correct amino acid. The anticodon is going to interact with a sequence in the amino acid sequence of the amino acyl tRNA synthetase. Valine, as you see right here, is going to be held in place, and an AMP holding it there. So this is bringing in the energy. This sequence right here is the only one that will interact with this sequence. So valine with this anticodon is going to have only one enzyme. Valine with the other codons that can also call for it, will have different anticodon interactions here for different tRNAs. So every anticodon tRNA here has its own synthetase. Even though there's some overlap, redundancy, because remember some of the amino acids can be called for, specified by more than one codon. Some of them have 2, 3, 6. So there's going to be 6 synthetases for that one amino acid. Each of those 6 is going to have a different codon-anticodon interaction there.